Hi guys, today we're talking about the factors and adaptations of photosynthesis. So first of all, you should have done a gizmo about the factors that affect the rate of photosynthesis. And before we did that gizmo, we talked about how color and the color of the light affects the rate of photosynthesis. So we saw that green light was mostly reflected by chlorophyll A and B. And because it's reflected and not absorbed, it's not used in photosynthesis, which means you're going to get very low levels of photosynthesis around the green spectrum. Colors like blue and red are absorbed by chlorophyll and they fuel photosynthesis. So you'll see high amounts of photosynthesis in terms of blue and red light if you shine those on your plant. In the gizmo, you should have noticed that blue was slightly higher than red. And so blue is typically the light bulbs for grow lights that you'll see if you were to buy a lamp for your plant to help it grow. Now in this lab, we were talking about the rate of photosynthesis. And to help us measure the rate of photosynthesis, we had to remember that photosynthesis gives off the gas oxygen. And so we can measure the rate of photosynthesis by the amount of oxygen being produced. And so oxygen is a product. And so as the rate of photosynthesis increases, you'll get more oxygen. And as it decreases, you'll get less oxygen. In other words, the faster the bubbles of oxygens are being produced, the faster the photosynthesis is happening. So let's look at some of the factors that influence photosynthesis that you saw in your gizmo. The first one you saw was temperature. Now the ideal temperature for photosynthesis is around 25 degrees or 75 degrees Fahrenheit, which happens to be that weather close to the equator. They have that year round, which is why photosynthesis happens a lot closer to the equator, and their plants are beautiful and amazing because they photosynthesize all year round. Now, at temperatures that are too high or too low, the rate of photosynthesis decreases. And at extreme temperatures, you will have a result of stopping the process of photosynthesis. So you should have noticed in your graph that it went up at first. It topped off. The rate of production was very high, around 25 degrees. And then after that, you'll notice a dramatic decrease. So why does it drop off so dramatically there? Well, that's because photosynthesis is controlled by enzymes that regulate the process. And enzymes, we talked about in the very beginning of the year, they are things that help speed up a reaction by hooking into the reactants and lowering the activation energy. But their shape is very important to them. And at high temperatures, their shape is destroyed. And we call that denaturing. So enzymes become denatured at extreme temperatures, changing their shape and their ability to speed up the reaction. So because you don't have enzymes helping, your reaction comes to a stop at high temperatures. And it's until you're able to replace the enzymes and get that process going again. So temperature, not too hot, not too cold. Make sure you record that in your notes. The next thing we looked at in terms of what affects the conditions or the rate of photosynthesis is light intensity. And light intensity is simply how much light hits the plant. So light intensity is like a dimmer switch. The brighter the light, the more light intensity you have. Maybe the closer to the light or the more lights you have, the brighter the light intensity. And as you increase light intensity, the rate of photosynthesis will increase. So you saw that in your graph, the rate increased the rate of photosynthesis. The more light intensity, the more you turned up that light, the more photosynthesis you had. But then it leveled off to a point. And at this point, photosynthesis is going as fast as it can and using up as much light as it can. So the extra light just is waste and does not get used by the plant. So more is better to a point. Also, notice that other factors will influence when we level off light intensity. So if you ran the lab with low levels of carbon dioxide, the light intensity would have leveled off lower than if you had high levels of carbon dioxide, where it would have leveled off higher. So carbon dioxide and light intensity kind of work together, so they will affect each other's graphs. Temperature will also affect this. At colder temperatures, notice that we level off earlier. At that perfect temperature, we level off higher. And as a side note about light intensity, the light intensity can be changed by changing your distance. So if you move the light source away from the plant, your light intensity will decrease, and that will decrease the rate of photosynthesis. All right, one last thing you saw in the lab that affects the rate of photosynthesis, and that's carbon dioxide. As carbon dioxide increases, the rate of photosynthesis also increased. And so you saw carbon dioxide go up, you saw the rate of photosynthesis came up, but then at some point, again, photosynthesis cannot work any faster and it will level off. So something else like light or water or temperature is limiting carbon dioxide, able to speed it up, and we level off the rate of photosynthesis. So again, with carbon dioxide, just like with light intensity, more is better to a point. So again, light intensity, carbon dioxide, and temperature, the three major things that affect photosynthesis. We could also talk about water. Water would also affect the rate of photosynthesis in the same way as light intensity and carbon dioxide, so in the same way. There are, of course, other things like the nutrients, the amount of chlorophyll, the amount of stress on a plant, disease. All of these things will also affect the rate of photosynthesis, but we can't directly measure those in our lab. All right, the last thing we're going to talk about are the adaptations for photosynthesis. The adaptations for photosynthesis are kind of interesting. Before we do that, we need to talk about a few things. So a stomata is a pore that allows water and carbon dioxide to come in and out. And it really allows the plant to breathe by taking in carbon dioxide and releasing oxygen. And guard cells are the cells that are around them that control whether or not the stomata is open or closed. So when temperatures are very hot, the vacuoles inside the guard cells deflate and the stomatas close, and that will help conserve water. So why do we talk about conserving water? Well, transpiration is a process of evaporation where water is evaporated through the leaves of a plant. And guard cells will close to stop the process of evaporation of transpiration. So a normal plant will bring carbon dioxide in, turn it into a 3-carbon molecule, and create glucose. It's a very efficient method. But sometimes during the day, those stomatas close and stop photosynthesis. But that's okay. It doesn't happen often enough. So in C3 plants, they're able to continue to live. But not all plants can do that. So they need to adapt in some way. So a C4 plant, these might live in a hotter situation. And so when the weather is hot and dry, the C4 plants close their stomata more than they would like. So what they do is when their stomata are open, they bring that carbon dioxide in and they store it in a closet as a C4 molecule. Then when they need to photosynthesize and they run out of carbon dioxide, they can just pull it out of this closet. It's less efficient because you don't get as much glucose out of this, but it allows them to close their stomata for longer periods of time during the day and conserve water. But if it's even hotter, then they can't open the stomata at all during the day. And these are CAM plants. So these guys can only open their stomata at night. So they do the same process as a C4 plant. They take in that carbon dioxide and store it in a closet as a C4 molecule, but they do that at night. Then in the day, they access that closet, they get that C4 molecule out, and they do the Calvin cycle. Also much less efficient. Now these would be more like pineapples or desert cacti, whereas C4 plants would be more things like crop plants like corn and sugar. All right, so that's it in terms of our different paths or adaptations. We take in carbon dioxide normally, we store it during the day for short periods of time, or we store it at night for long periods of time so we can keep our stomata closed when we need to. Let me know if you have any questions. Make sure your notes are complete and complete the exit poll on this topic.